see if it's working. Sebab tadi actually saya online guna laptop daripada fakulti, tapi not sure what happened. So saya transfer ke desktop, so tak tahulah if ada yang berubah ke apa ke. So anyway, Assalamualaikum and very good morning to everyone. So I hope you are all doing well wherever you are. You are still at home, right? Ke kat lab?
College.
College. Oh, okay. All right. So today's lecture will be on the laboratory investigation for infectious diseases. Okay. So I think from the previous lecture, you had already understood what infectious diseases are all about, the examples of infectious diseases. Okay, so today we are going to look into what are the tests that we perform in the lab, particularly for the diagnosis of infectious diseases. Okay, I hope you can stop me whenever you feel that the line is not okay or something like that. Okay, so jangan biar sampai the end of the lecture baru cakap line tak clear. Okay. All right. So next. Okay. So what is infectious disease? Hafiz Iqbal, apa infectious disease?
Disease yang boleh menyebabkan jangkitan.
Okay, so it is a disease, okay, in which this type of disease can be transferred from one person to another, and it can be through the vector. Okay, vector ni maksudnya animal lah, okay, ataupun it can be through sentuhan dan sebagainya lah. Okay, so that is what defined as infectious disease. Okay, so apa contoh infectious disease?
COVID-19.
COVID-19, okay. Lagi? Ada lagi contoh lain?
TB.
TB, okay. So TB ni it can be transmitted through droplets, sama juga macam COVID lah. Okay, but TB ni dia lebih kepada jangkitan paru-paru. Okay, alright. So when we talk about the diagnostic cycle, there are 3 different stages. Okay. So the first stage is the pre-analytical, second stage is the analytical, and the third stage is the post-analytical. Okay, so if under the pre-analytical, it includes the sampling. Okay, sampling ni maksudnya when you when there is an outbreak, okay, so we take the sample from the patient or the person under investigation. Okay, the sampling can either be from the stool, it can be the swab sample, okay, or even the blood sample. Okay, so those are the pre-analytical. And then the labeling. Okay, labeling is very, very important because if there is a mistake in the labeling, then the results will be not reliable. Okay, so a person who is healthy could be, could be diagnosed with COVID-19 or TB, okay, due to the wrong labeling. So this is very, very crucial, okay? And the transportation of the sample. So from one place to another. So let's say the sample is taken from probably water sample or lake. So you got to bring that sample or specimens to the laboratory. Okay, and to do that, there is a specific containers, bottles that is needed to be used. Okay, you cannot simply use the, just whatever you can find. You cannot use that. Okay, because for lab diagnosis, it is very, very crucial. Okay, and even the transportation and storage is, it will determine whether the results is reliable or you might get false positive or false negative results. Okay, and also the specimen clinical information. This is another important aspect of the pre-analytical stage because once the sample arrive in the lab, then there should be enough or accurate information about the type of sample and also what test is requested. Okay, So let's say the sample is taken from the blood. So once it arrives the lab, and the nurse has to direct or order very important tests. So it has to be very, very specific tests. Okay, you can't say, okay, do whatever you want with the sample. You can't do that. Okay, it has to be very specific. And then for the analytical, which we will learn further for today's topic, is how we identify the microorganisms, okay, that leads to the infectious disease, okay, and how do we interpret the results. So this is very, very important because sometimes we do not know what actually caused the infections. So we would need to perform the identification, okay? And if I talk about microbial, then it doesn't mean bacteria. It can be virus, it can be fungus, even parasites, okay? So post-analytical is where the analysis is already completed and the final report is given to the physician. So they will report to the patient or to the person under investigation. Okay, so this is the post-analytical. Okay, so if we talk about the microbiological specimens, macam nak remove ni. Okay, if we talk about the microbiological specimens, first, it is about collection and preservation, okay? And the laboratory responsibilities is where once the sample arrive in the lab, prior to any analysis, so the lab personnel has to make sure that the specimens that they receive is the right specimen, okay? And the amount is enough for whatever analysis that is requested or ordered. And also the information that is attached together with the sample is accurate, okay? Because sometimes, you know, now we know about these COVID-19 cases increasing from day to day. I actually personally, I involved in COVID-19 lab. Okay, so we actually carried out tests, the RT-PCR test in HPUPM. So what happened is that sometimes the result can be delayed because we receive forms with the same name, okay? So, but different. Tak dengar ke suara saya? Hello? Ada problem dengan line ke?
Umi.
Can you please turn off your mic, please? Your mic. Admin boleh mute kan? Host boleh mute kan?
Betul.
Busy ni Umi ni. Okay. Okay, so the laboratory responsibilities. Okay, so need to make sure the sample is the right sample and also the form that is attached together with the sample is correct. Okay, because I mentioned to you just now, the result can be delayed because we receive the same different specimen but the same form, meaning one form for two specimens. So we need to, you know, trace back from where the sample is collected and which form is the right one. And at certain extent, we will reject all these different samples like this, okay, because we do not want any problem. And then the test requisition is what test is requested. And also together with this test requisition form, there will be information about the specimen that is collected, okay? And then after we approve the information provided to us, we will start process the sample, okay? And in the processing stage, we will first verify the quality of the sample, all right? So if the sample is taken 2 or 3 days back, okay, sometimes it can affect the results. So we do not want that to happen. Okay, so that's why we need to verify the quality of the samples that is received, all right? And also if there is a case like the same name with the same sample, we will reject the samples. Even if the amount is not enough or, you know, the test requested is blood analysis, but we receive stool sample. So we will need to reject the samples, okay? And also handling of samples. Some sample like urine, if it is taken a day before, it needs to be stored at 4 degrees Celsius. Okay, so if it is not the case, then you know what happened is that we will get more and more bacteria growing in the urine sample, so it will affect the result as well. Okay, so under the processing, first we need to verify, reject the sample if there is a problem with any of the important issues, and also we will need to look into how the sample is being handled before it arrived the lab. Okay, and also storage and disposal. So most of the time after the analysis, after we process the sample, we can either store it for future research, or we can also dispose it. Okay, in HPUPM, we actually have thousands of COVID-19 swab sample. Okay, that is because we will use it for research. Okay, so whether we will need to store or we will dispose it, it depends on whether we have the ethics to store it. And if we don't have it, then we need to dispose it straight away after the processing. Okay, and also the transportation. It depends on what specimens that is being collected. There are different methods of transportation. Okay, so apa tadi? It's not moving. Okay, all right. So this is how the patient suspected with infectious disease, the sample is being analyzed, okay, in the lab. Okay, so this is, let's say this is a patient with suspected infectious disease. We can use the microbiological route or the immunological route. Okay, if we are looking for the microbiological route, then these are some of the samples that should be collected. Okay, it can be blood, feces or stool, urine sample, tissue biopsy, mucosa swab. Okay, so these are the samples that will go through the microbiological route. And if you're looking for the immunological route, then we will take or we will collect the blood sample. Okay, and the assay or the test that will be performed is also different if you're following the microbiological route or the immunological route. Okay, so under the microbiological route, there are conventional microbiology or the molecular biology approach. Okay, so the conventional is where we just grow the bacteria, or let's say we receive the sample from blood. So we will try to recover bacteria, we will try to recover the parasites or fungus from the samples. Okay, so we will use a medium in the lab. Okay, so I will show you what medium is all about. And also following the pure culture isolation, we will perform the identification process. Okay, and if you are following the immunological route, then the test that we will perform will be the antibody or antigen assays. Okay, it depends. And these are some of the examples of immunological tests. Okay, and for molecular search, normally molecular microbiology is used for confirmatory analysis. Okay, so let's say you've got something here that is positive. Sometimes we will perform the molecular analysis to confirm the results. Okay, or sometimes the bacteria, the fungus, or even the virus, it is not possible to be grown in the laboratory. So what we will do is we will perform molecular biology approach straight away. We will perform PCR or RT-PCR. Okay, so it depends on what bacteria we are looking at, and also whether we are using the immunological route or microbiological route. Okay, so that will determine what tests we will perform in the lab. Okay, so in order to identify the microbial pathogens, okay, there are three different methods that can be used. All right. So for your information, I will go through every specific infectious diseases examples of what test is being carried out at the end of the lecture. But I think it is very important for you to first understand the basic of the lab investigation. What the test that is being carried out in the lab, what are the gold standard, okay? So you will need to understand the basic first, then you will be able to digest what I will explain to you at the end of this lecture. Okay, so the phenotypic method is where you observe the bacteria macroscopically or microscopically. Okay, macro means you just use your eyes and you'll be able to see the bacteria or fungus or the virus, okay? But for microscopic, you will need to perform maybe a staining technique, okay, in order to observe the presence or detect the presence of bacteria. Okay, and also there is a biochemical test that can be done, particularly to identify the pathogens. All right, so immunologic is where you perform the serological analysis. And genotypic, I mentioned to you just now, this is normally used as a confirmatory test because it is very specific, it's very sensitive, okay? And this will involve the genetic or DNA, RNA, and also molecular techniques. So there are many different genotypic methods that normally we will perform for infectious disease. Okay, so we will look into what phenotypic methods is all about. Okay, so I just want to know, are you in second year now or first year, second semester?
Third year.
Third year.
Oh, third year. So you have gone through microbiology, is it?
Yes.
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 With me?
Yes.
Okay,
Yes.
okay. All right, okay. So I think because you had covered all these different microbiological analysis in microbiology subject, so you should be able to understand better. Okay, okay. So you know microscopic identification is where we observe the bacteria or the pathogens on the agar plate. Okay. So different morphology, different characteristics, different pigmentation produced by the bacteria will determine their species. Okay, so for instance, if you have Staphylococcus epidermidis and Staphylococcus aureus, so you should be able to differentiate based on the different color that it produces on the agar. Okay, and this is very, very important because sometimes when there is an outbreak, okay, this outbreak can also leads to co-infection. Okay, so I'm giving you an example. So there is one patient with COVID-19 positive. Okay, so when we perform DNA analysis, we found out that the bacterial DNA also present in the sample. Okay, and because of that, that particular patient is considered as having co-infection, meaning on top of having a viral infection, COVID-19, he also had bacterial infection as you know, together. Okay, so for that we will need to perform a test or culture like this if the bacteria can be isolated, can be grown in the lab. Okay, not all bacteria can be grown in the lab. Okay, so it depends on whether there there is a possibility of that bacteria of growing in the lab or not. So we can perform this type of analysis. Okay, so this is an example of two different species observed on nutrient agar. So we have epidermidis having a very, very tiny colony, and also E. coli, which is a little bit bigger, okay, in terms of the size. And some of the bacteria like Pseudomonas aeruginosa, they produce green coloration. Okay, so sometimes when we take the blood sample from the patient and we observe, many different colors of colonies grown on the agar. So if the bacteria produce green color like this, so we know that, so in the blood sample, there is also Pseudomonas aeruginosa. In the sample, we also have Staphylococcus aureus and so on and so forth. Okay, so blood is supposed to be sterile. So if you detect blood, if you detect bacteria or virus or fungus in the blood, so what does it show you? So maknanya you ada infection lah. Okay, either septicemia or bacteremia. Okay, and also bacteria like Serratia marcescens also produce red coloration. Micrococcus luteus. Okay, so this is one of the method that is always we will perform in the lab in order to identify the bacterial pathogen. Okay. So this one, I think we had covered during the microbiology lecture before. So this is very, very crucial as well, although it can be, it varies, okay, from one eyes to another. But some bacteria, they have very distinct structure, distinct characteristics that make it easy to be identified or differentiated from one species to another. Okay, and also if you are using the slants agar, so this is how it will looks like. And if you're using culture, so you will see a different turbidity with different oxygen requirement. The bacteria will grow either at the tip or the edge of the culture or at the bottom. So those are the different analyses that can be performed in order for us to identify the microbial pathogens. Okay. Okay, so this is very familiar to you, I believe. So we have blood agar. So blood agar is used mainly to identify bacteria. Because I mentioned to you, in our blood, no bacteria should be detected in the blood. Okay, but if you can detect it, so you have infection. And sometimes it can be very tricky to isolate bacteria from the blood. Okay, so that's why we use blood agar, which is an enriched agar, in order to support the growth of the bacteria. Okay, and if we are performing a comparison between two different species, we can use McConkey agar, and this is mainly for gram-negative bacteria. Okay, so no gram-positive bacteria can grow on McConkey agar. So if you suspect that the patient or an individual has infection due to gram-negative, so you can opt for McConkey agar instead of the blood agar. Okay, and if you have a case where there is an infection caused by Haemophilus influenza, so you can use chocolate agar, because this is another enriched agar, but it is mainly for fastidious organism. Okay. And then we also have clad agar. Clad agar is also another common agar that we use in the lab. And if you are performing antimicrobial susceptibility testing, mainly to help or to assist in the treatment, okay, antimicrobial treatment, that you can use this type of agar. And also alkaline peptone water, sabrodextrose agar, mainly for fungus. Nutrient agar, if you don't have any other agar to grow, then you can always opt for nutrient agar. And Lowenson-Janssen medium. Okay, so this is another agar that we normally use in the lab to identify the bacteria. Okay, so all of these are the microscopic observation. Okay, and then this is all about the microscopic identification, where in order to understand the principle of all the staining techniques that we perform in the lab, then you will need to understand the structure of the bacteria. Okay, so this is mainly for bacteria only. All right, so you know the difference between gram-positive and gram-negative. I hope you can recall. Okay, so the difference is in the peptidoglycan layer, whether it is thick or thin. All right, and that will determine the appearance at the end of the staining technique. Okay, so we have simple staining. Okay, so simple staining is just using one type of reagent or color reagent, in which the reason why we perform simple staining in the lab is just because we wanted to know whether the bacteria that cause the infectious disease is a cocci shape or is it a rod shape, and also in terms of the size of the bacteria or the pathogen. Okay, and this is very, very important, because sometimes we suspect that the bacteria that cause the infection is E. coli, but after the staining technique, actually it's gram-positive, not E. coli, which is gram-negative. Okay, so there must be something wrong probably during the transportation, or there is a contamination during the process of sample collection and until it arrived the lab. So those needs to be clarified. Okay, and also we have gram staining, in which we stain the cell wall of the bacteria. So all gram-positive will appear as blue or purple. And also gram-negative will appear as red due to the discoloration by alcohol. Okay, and also we have endospore staining. So let's say there's a case where there's an outbreak, okay, food poisoning outbreak probably. So a group of people got diarrhea because they went to eat at a stall, okay, and it is suspected that the bacteria that caused the disease is Clostridium species. So what you can do is you can perform endospore staining. Okay, but normally food poisoning can be caused by Salmonella species. So Salmonella is a gram-negative, so you can perform the gram staining. But if the infection is caused by bacteria that has endospore, you can confirm that by performing endospore staining. So this is how it looks like. Okay, and also if we receive, the lab receive sample from sputum, okay, suspected of TB case. So the best staining technique that can be performed is the Ziehl-Neelsen staining, where we stain the acid fast bacilli. Okay, so this is how it will look like. Okay, and then we can also perform flagella staining. So flagella staining is only for those bacteria that can motile. Okay, so if we suspect that the infection is because of E. coli, so you suspect that, maybe we can perform the flagella staining just to confirm, and then you can perform this. Okay, and also capsule staining. So if there is a case of meningitis, so you can perform capsule staining. So you can see the presence of capsule at the end of the staining technique. Okay, and also diphtheria. Okay, diphtheria. Normally, we will perform Albert stain, where you stain the metachromatic granules. Okay, it appears as purple, dark purple color, and the cells will appear as green. So this one not to be confused with the endospore staining. Okay, even the method is different. Actually, there's a video here that you can use, so that you can watch. I will also give you the link later. Okay, so you can watch how it is performed, and it is very, very important to select the best staining technique for different disease. Okay, so you do not want to use wrong technique for if you are suspecting a gram-negative and you perform Albert stain, that is not the right analysis. Okay, and then we have biochemical test, which is also a macroscopic analysis. Okay, this one you can see based on the reaction, okay, of the sometimes bacteria, when they grow, they will produce certain enzymes. Okay, so we manipulate that by performing this biochemical test. All right, so for gram-positive bacteria, we will perform tests such as catalase, coagulase, MSA. Okay, we grow the bacteria on mannitol salt agar, and also CAMP tests. Okay, and gram-negative examples are like oxidase test, MR repeats test, Siemens citrate agar, and also urease test. So this, I think, also had been covered during your microbiology lecture. So catalase test is where if we suspect that the disease is caused by certain species, and we know that this species, normally it will produce catalase enzyme. So you can perform this to confirm that the species that you observe on the plates is the species that produce the catalase, that specific species. Okay, and how it is done, sometimes because of catalase production, that it would form bubbles. It depends on the technique as well. Sometimes even if you apply, you can't really see the bubble, so it is very technical. Okay, and also coagulase test, mainly for staphylococci species. So you want to differentiate between coagulase negative staphylococcus and coagulase positive. Okay, so you can perform this coagulase test. All right, so you have mannitol salt agar test over here, where you will differentiate between the two different species. Okay, and the reason why Staphylococcus is able to grow on this mannitol salt agar is because it can tolerate high concentration of salt. Okay, so this agar is prepared with high salt concentration. Okay, and also CAMP test. Oxidase test, okay, so you looking for bacteria that produce oxidase, and also MRVP test, okay, methyl red Voges Proska test, okay. So these are all the indicator and also a confirmatory test that can be performed in the lab to confirm the species of the bacteria. Okay, Siemens citrate test mainly for Enterobacteriaceae. All right, so if there is a case of food poisoning or diarrhea, and we can perform this Siemens citrate test, and also urease test where you observe the presence of urease enzyme. Okay, so positive will give you pink color, and negative will not change the original color of the test tube, the test agar. Okay, and in the hospital, normally we will have an automated system of biochemical tests in which we have like a card. So this card comes with a different panels of biochemical tests. All we need to do is just inoculate the bacteria, okay, on this test card, and then we put inside the machine like Vitek, BD Phoenix, okay, Microscan. So these are all the different machines that can help us in identifying the species or confirm the species of the bacteria based on its biochemical reactions. Okay, so in the hospital, we don't have much time to actually perform single biochemical tests like what I had presented to you just now. So normally we will have like an automated system like this. Okay. Okay, and then we move on to the serologic methods. Okay, serological methods. So for serological methods, there are two different methods. It can either be direct serological method or indirect serological method. Okay, so for direct serological method, normally this is used for identification of unknown microbes. Okay, and for that, we will detect the presence of antigens. Okay, so we know already that this bacteria or this virus, it caused this disease. So we have specific antibodies for that particular pathogen. So we use that to detect the presence of antigens. Okay, and normally this direct serological method is used for serotyping and serogrouping. Okay, serotyping or serova is a distinct variation. So you have a species, but it can be a different variation of its antigen identity. Okay, so that is called serotype. And a group of serotype or serovars is called serogroup. Okay, and indirect serological method is where you detect the specific and non-specific antibodies. Because you know, when there is a bacteria or virus or fungus or parasite, then you will be able to detect the antigens. Okay, so if you detect the antigens, meaning you can, you know that in that sample there is microbes. Okay, but for indirect serological method, you will detect the antibodies. Okay, in this case, we will detect IgM and IgG. Normally we will have antigens or the organism itself. So IgM is normally an active infection, and IgG is when you already had the infection. So IgG will remain in your body. Okay, so that's why back to the case of COVID-19, sometimes following quarantine period, right? So they will perform RTK, okay, rapid test kit, right? And this sometimes give you IgG positive, sometimes give you IgM positive. So if it's IgM positive, then we know that the bacteria is, the virus is still, you know, the number of virus is still high in the blood sample, okay? And if we only detect IgG, it means that the body has started to form more and more antibodies towards the virus. Okay, so that's why post-infection, normally IgG, active infection can be IgM. And there's also case where one of the patient already positive with COVID-19. So what we detect in the blood sample is IgG. So it's past infection. But after some time, he was again, you know, he came back, and we detect IgM. So meaning he had a second exposure. Okay, so this is an indication of whether that patient has active or post-infection. Okay, and these are common immunoserological tests. Okay, but there are more. The list is a lot. Okay, but I can just highlight to you few. So there are hem agglutination, EIA or enzyme immunoassay, latex agglutination, immunofluorescence, antibody test. So depending on what you're looking for in the pathogen, so you will be able to use a different serological test. Okay, so which test to be used normally, yeah, like I said, it depends on the disease and also what pathogen you collected lah. Okay, all right, so we move on to the next one, which is the molecular method. So this molecular method is very important, okay, especially if the bacteria, the virus or parasites, fungus, they cannot be grown in the lab. Okay, some bacteria you cannot grow in the lab. Okay, not every bacteria you can grow in the lab. So that's why if we have case like this, what we will do is we will go directly to the to the molecular methods. Okay, and why we are able to perform the molecular methods is because every organism contains unique species-specific DNA sequences, and from there we can do identification, okay, in order to know what species the pathogen is. Okay, so different stage of this is a central dogma of biology. Okay, so if we extracted DNA from the pathogen, then we can perform PCR, we can perform PFGE or pulse field gel electrophoresis, we can perform multi-locus sequence typing, and also microarray. Okay, that is when we extracted DNA from the pathogen. But if we extract the RNA, then we can also perform PCR, but we will need a different technique in which we will need to translate this into cDNA or complementary DNA. Then only we carry out the normal PCR cycle. Okay, and on top of that, we can also perform northern blot technique. And if we have protein, then we can perform ELISA, okay, and also Western blot analysis. Okay, so for PCR, this is a method that is where we try to imitate the natural DNA replication. Okay, so it is an artificial process. So we will amplify more and more of the specific DNA. Okay, and at the end of the PCR cycle, we will get billions copies of that particular DNA. Okay, so this is DNA. We don't amplify the entire DNA. We might need just, this whole lot. So we will amplify this whole lot until it becomes billion copies. Okay, and it can then be detected. So these are the different types of PCR, standard PCR, multiplex PCR, RT-PCR, and also QRT-PCR. Okay, this is the test that we normally perform for virus. Okay. So this is the principle of PCR. From DNA, we are looking for a specific template or wanted DNA, okay, or DNA of interest. The first cycle, it will amplify into 4, okay, 8, and then 16, 32, and so on and so forth. Okay, so this number of cycle can vary, it varies. You can perform in 20 cycle, you can perform in 30 cycle, it depends, okay? And at the end of the 36 cycle, you will get 68 billion copies of this small piece of DNA. Okay, then you should be able to detect it using the gel electrophoresis technique. Okay, so this is what you will observe on the gel electrophoresis. Okay, following the PCR, you need to confirm that the DNA is there. So you will perform a technique called gel electrophoresis. And this technique allows you to determine whether your PCR cycle is successful or not, or whether the DNA is detected in the sample or not. Okay, but this is conventional method. There is another advance where you can observe the presence of the DNA without performing gel electrophoresis. Okay, so for multiplex PCR, so let's say we have sample that we suspect it can be multi-species. So we can perform multiplex PCR. So if you detect more DNA like this, so you know that in that particular sample, there are more than one species of pathogen, okay? And also real-time PCR, maybe now you are very familiar with this because COVID-19 RT-PCR, COVID-19 RT-PCR, right? So this is what we actually monitor in the lab. So let's say this is the DNA, the cDNA, okay? So the DNA will be fluoresced, so we will be able to detect the presence of the DNA straight away. It is a live process, okay? You don't have to perform gel electrophoresis. So you can just use this RT-PCR, and at the end of this cycle, straight away, you know whether the DNA is present or it is absent, okay? And on top of that, you can also quantify, okay? So let's say this is a sample from an outbreak. So you wanted to know whether the viral RNA is detected. So what you will do is you will perform the RT-PCR. So let's say you detect the peak somewhere here. What does it mean? So it means that the viral load is high. Okay, if it goes until here and there's a peak over here, then you know that the viral DNA, viral RNA is low. Okay, the viral load is low. All right, so basically this will determine whether, sometimes it can determine the stage of the disease. Okay, whether it is at the earlier stage or it is at the peak where you can detect the RNA before the positive control peak goes up. Okay, or you can detect it towards the end of the PCR cycle. So that can be considered as low viral load, okay, or low bacterial load. So we also have PFGE technique. PFGE normally used for DNA fingerprint. Okay, so normally if we have an outbreak from different places, so we wanted to know how one sample or one disease is related to another. So we will perform this PFGE, all right, and towards the end of the analysis, we will look at this dendrogram, and the different band observed, like this one, right? And this one, this is related. So this one and this one might not be related. So this closer to this one. So something like that. There is a technique to analyze the results, okay? So this dendrogram is very important because it determines whether sample 1 is related to sample 2 or it is related to sample number 6. Okay, so let's say this is from Pahang, this is from Johor, this is from Selangor. So we know that sample from Pahang and Johor is related, but sample from Selangor has, although it is, you know, there is some similar identification, but still it is not closely related. Okay, so you will need to perform this in order to know how they are related or how they can be distinct from each other. And also MLST. So MLST is another molecular technique that can be carried out, but it is limited to the organisms that is in the database. Okay, so if the infection is caused by a new or novel species, then you might not be able to perform this MLST analysis. Okay. And also microarray, where you detect the presence of protein, all right? And sometimes when the bacteria or virus or any pathogen cause an infection, they will express certain level of protein or enzymes. Okay, so this is a technique where we use to observe the presence of those particular protein or enzymes. Okay, it's called microarray technique. Okay, so now I will go through with you these lab investigations for vector, food, waterborne, and STD. Since you already know what are the different techniques carried out in the laboratory, you know, particularly on the principle of the test, and you're familiar with all the different types of tests that is carried out. So now we can focus on the different diseases caused by different organism, okay? So let's say we are talking about vector-borne diseases. So you can see that if the vector is mosquito, different species of mosquito can lead to different disease, okay? And from there, we should be able to identify the type of pathogen, either that infection or that disease is caused by virus or parasite or bacteria. Okay, so aquatic snails, black flies, fleas, lice, sand flies, all this will lead to different disease, and we need to know the type of pathogen because this will determine what test we will perform. Okay, so these are another sets of vector-borne diseases. I'm not sure whether this had been covered before. I think some of it had been covered, right? So what I would like to highlight is when you talk about vector-borne diseases, you know it is caused by vector, okay, or insects. And important for us to know the type of pathogen that leads to this infectious disease, because if it is caused by virus, then you might not use the culture method. Okay, so you need to opt for probably the serologic or the molecular technique. Okay, so this is the different lab investigations for vector-borne diseases. I don't have time to go through with you one by one, but you can read further about this. Okay, so if we're talking about the conventional or the classical method, it can be microscopy based on the presence of, when you take sample like blood, tissue smear, so you can perform microscopy examination. Okay, and also pathogen isolation using cell culture. If you're talking about the serology-based method, we have IFA, immunofluorescent assay, we have ELISA, immunoblotting, PRNT is a serological test. This test, it uses specific antibody to neutralize a virus, okay? And SN is a serum neutralization test or plaque reduction test. This is used to confirm the identity of a virus, okay? And we have hemagglutination inhibition test or HI. This is also a test that we use to titrate the antibody response if there is a viral infection, okay? CFT is complement fixation test. And this test is a test that we perform either using specific antibody or antigen, and we use identification from serum sample, okay? And these are the omics tests, NGS, okay? Next generation sequencing technique. And we also have multiplex methods like microarrays, multiplex PCR. Okay, so these are the different PCR techniques over here. So the normal PCR, RFLP, which is the polymerase chain reaction, restriction fragment length polymorphism. Okay, so you can read further about that. RT-PCR, very common. FISH is fluorescence in situ hybridization. LAMP is loop-mediated isothermal amplification. Okay, so these are the different techniques that can be performed. It depends on what is the end result that you wanted. Okay, so that will determine what test you will use. And then if you're talking about the food and waterborne diseases, so these are the organisms that leads to food and waterborne diseases. And if you're talking about food and waterborne diseases, normally it will, it is all isolated from the stool specimen, okay? And these are the different tests that can be performed. And different tests, normally it is meant to detect different types of bacteria or parasites, okay? And you can read further about this, all right? So if the sample that is collected from the patient is blood, then we can perform the full blood count, all right? So particularly to just look whether we can detect high white blood cells amount in the blood, full blood count analysis, and that will lead to, a sign of bacterial infection. And if we can also detect the presence of bacteria in the blood, then it is probably due to the disease caused septicemia. Okay, and stool antigen test, also another test that we perform for specific bacteria or Hep A, hepatitis A. Okay, and then, oh my God, I have another class after this. So this is sexually transmitted disease. Okay, so we have chlamydia testing. These are the different specimens that can be collected. And normally this chlamydia testing, it detects the genetic material, the DNA of the bacteria. Okay, Gonorrhea testing also detects bacterial DNA. Hepatitis B, this is from virus. So we can perform ELISA, or if we want to perform PCR also can, in which we will detect DNA instead of the antigens or antibody. Okay. Hepatitis C testing also can be performed using molecular techniques or the serological methods. And we also have herpes testing, HIV, HPV tests, okay, and also syphilis. Lastly is the trichomonas testing. So this trichomonas testing is for infection called trichomoniasis. Okay, so this trichomoniasis is a sexually transmitted infection, and it is caused by parasite. Okay, so for detection of parasite, we can either perform PCR, or we can also perform microscopic observation. Okay, so to conclude our lecture today, when we talk about diagnostic cycle, it consists of pre-analytical, analytical, and post-analytical. Okay, it's very, very important, because if the pre-analytical is, is not good, then the analytical results will be affected. Okay, and different methods that can be used for identification of bacterial pathogen in the lab. Either we use the phenotypic methods or immunological analysis. And for confirmatory, normally we will perform the genotypic methods or the molecular approach. Okay, and all in all, the selection of tests for diagnosis of infectious disease, it depends on the type of pathogens, okay, and the infection that they cause. So you will need to first know what infection caused by what pathogen. Then from there, it will direct you to what tests that should be performed in order to analyze the bacterial or microbial pathogens. Okay. Okay. All right. I have another class at 11, so I have to leave. But if you have any question, can you email me? Let me just. Are you all okay?
Okay.
Are you all following me?
Yes.
So basically, there are so many different tests that we perform in the lab. But like I said, the principle is first you will need to know what outbreak that is, what infection that is. And from there, we need to either perform the serological methods or the phenotypical method. Okay, and this is actually depends on whether you just want to do the screening. Okay, so you just want to see whether the bacteria or the microbe is present. And if you want to know exactly what species is that, then molecular technique is the best approach. Okay, so I have to leave now. Please email me if you have any question. I will pass this lecture notes and also the recorded session nanti. Iqbal Hafiz ni email everyone Hafiz, are you there?
Okay.
Okay. And I will also email to you the lecture notes and the link, okay, all the related links to this lecture. So if you have any question, email me. Okay, thank you. All right, so I'll see you later. Maybe next time, okay? Actually, I'm in the faculty. So nak belanja makan sebelum puasa kan? Maybe next time, okay? Please stay safe and see you when I see you. Take care. Assalamualaikum.
Thank you, Dr.
Have a nice day.
Day too.